And now I'm delighted to welcome Emmanuel Lourie, who is a PhD student at the Movement Ecology Lab at the Hebrew University of Jerusalem, where they use the Atlas system to track the highly detailed movements of fruit bats. And today he's going to be talking to us about spatial partitioning between neighboring wild fruit bat colonies. And we're very much looking forward to your talk, Emmanuel. So over to you.
Thank you very much, Lisa. It's very good to be here. They're not here, but I want to also thank Macy and Rachel for organizing this. It was a pleasure communicating with you guys. And yeah, before I just share my presentation, I kind of have to say that I'm particularly excited to be here just because essentially I'm the only researcher working on bats in my lab. The other people in the Movement Ecology Lab are working on birds. So it's good to be amongst other bat lovers, and I would really appreciate any type of input or feedback that you might give, because it will probably be very different from what I'm used to. So yeah. I'll try and share my screen. Elisa, just please let me know whether it works fine or not. Let's see. Okay, can everybody see that?
Yeah. Yes, we see your slides.
Okay, good. Okay, so thanks again. And today I'm going to talk on one major topic, which is kind of contradicting this NGO's title, and to show you that bats actually have some sort of borders, and more particularly, I'm going to talk about spatial partitioning between neighboring fruit bat colonies here in Israel. But because we have some time, I also want to take this opportunity to kind of take you through the process of how this question even came to be, and also what types of other research questions we're asking after we saw these results, with some preliminary results that I will be happy to share with you. So the general topics that we're going to follow would be, first, I would like to introduce you the Atlas tracking system. It's a very unique system that we love and serves us very well. And there's a few things that is worth considering before you actually see the bat data of their movement. And then we want to go through looking on our exploration of individual differences in movement, which are very stark in this particular species, and the things that maybe can or cannot explain these individual differences in movement. And then we're going to move to the major topic of spatial partitioning. And finally, if time permits, maybe I can share with you some of the cool field experiments that we've done to try to understand if and how information is transferred in the species. Okay, so starting off with the Atlas tracking system. So what you're seeing here on the map is basically one night's worth of movement of one single bat that we tracked for, in this case, every 8 seconds. And this is possible thanks to a very unique system that works very similar to how GPS data works, only that instead of the tag communicating to a satellite in space or to several satellites in space, it communicates with a set of an array of antennas that are spread around the study area. And what the main benefit of this is that the localization that you get each point here on the map is actually calculated on the antenna side, on the receiver side, and not as it is on the GPS on the tag itself. What this actually allows is that the tag is less energy demanding because it doesn't have to do all this complex calculation on its own. It's much lighter and cheaper owing to that. But of course, because it's based on antennas that you decide to spread or that your lab decides to erect and to buy, then of course it's limited in its spatial coverage. So not very good for to study full migration tracks, but excellent for our species, Rosetus aegyptiacus, which really spend most of its time in a limited home range. We recently published an article in Science that compares all those different technologies. So you will be probably familiar at least with 3 of those. So on the top right here, marked in green, there's manual triangulation. So the old school way of going with an antenna in your hand or your Jeep and trying to locate an animal you tagged before. So of course, you won't get many data points because it really depends on how well you can find it, and the localizations themselves are not going to be particularly accurate. And so we deem it also not super cost effective, not because it's not cheap, because the technology is cheap, but because you can't really get a lot of data from each US dollar that you invest, really. On the other end, there's the GPS logger and GPS with uploads. So the GPS logger just stores information and it gives us much better temporal resolution. It's also relatively cost effective, even though they cost a lot. The problem with the loggers is that you'll have to go and find them if you want your data. So you have to know where potentially it can be dropped off. Or you can go for GPS with the remote download, normally through a GSM system or through the satellite itself. But then the tag itself will be heavy, much more expensive. And of course, if you have this tag, you would probably not use a very high temporal resolution because you will want longer duration of the tag itself. And if we're talking about bats active at night, then you can't even add to it a solar panel that will actually help the battery survive for longer, then the investment is even less effective. In comparison, reverse GPS system, which Atlas is one of them, has better temporal resolution. So we can track animals even every 1 second. And it's 3 to 5 orders of magnitude more cost effective than its GPS counterpart, the same size of tag, if you look at the GPS. And of course, because it's cheap and light, we can track many individuals simultaneously without being background. Okay, but so that's nice in terms of technology and investment, but what actually can we gain scientifically, right? So first of all, and maybe most important for us, the number of species that we can track and follow are much bigger just because the tags are so light. So we can track even small species, and we have examples of studies using reverse GPS system, particularly Atlas, on noctules, on Culis pipistrelle. Anyways, you can track individuals that weigh up to 10 grams, which is more or less 63% of all extant species. With the GPS as comparison, you can basically track less than 20%, the big heavy ones, if you can catch them. In terms of actual research questions, then of course, if you have high resolution data, you'll get more potential to answer more questions. So just a schematic example here, artificial tracks. So if you have high resolution data and you want to, for example, ask whether bald individuals versus shy individuals are more or less explorative. So you would take different birds, you would assay them in your lab, and let's say that you have a dichotomic, unrealistic, but still bald and shy individuals, and then you tag them and you let them fly. So if you track them in 5 seconds intervals, as you can see here on the left side, then you would notice that one of them is very explorative, going all around and zigzagging. This is in dark blue, and the bright blue one goes straight ahead, is much less explorative, and you can say that the bold individuals are more explorative than the shy ones. Great. But if you subsample this exact same tracks and, I don't know, get a location every 30 minutes, which is very common, then those two birds look like they behave in the same way. And then we lose our inferences. We can actually even conclude the wrong thing, right? And it also, there are other examples for this. For example, if you're interested in interactions between individuals, then you can see on the left side, we can identify at least 4 interactions where maybe information or diseases are transferred. But on the right-hand side, it looks like they don't interact, which obviously is wrong. Okay, let's talk about more specifically about this species of study. So we are studying the Egyptian fruit bat. And as you can see from this picture, this is a very typical way of seeing them. This is a Ficus microcappa tree in a small city in Israel. And you can see plenty of bats foraging on it in huge numbers. So it's a very abundant species in Israel. It can reproduce even twice a year, and they often do. It roosts almost entirely in caves and buildings, very, very, very rarely, to the extent that almost never, we see them roost on trees like other fruit bats do. And as you saw from this video, it's very gregarious. We can find tens to thousands in cave roosts, some of them are huge, and many on trees, even more than 50 that is written here. You can even see it on the video in the back. It's very generalist, so it feeds on a huge variety of fruits, basically everything that has sugar, and is available, and you can land on the tree and eat it happily. And it's the only fruit bat that is distributed behind the tropics, to the best of our knowledge, which could maybe explain why it is specialized in roosting in caves and buildings, and also maybe could explain, I'm not going to go into it, but why it has very specific echolocation abilities that other fruit bats don't, using its tongue, but we won't get too much into it. And maybe the most important thing, it's cute, and I don't think I need to explain why. So for this study, for the special partitioning part, we tracked 107 bats during 2018 and 20. To catch them, we used mist nets, basically classical mist nets used for marine birds are fitting to our needs. We did it around fruit trees mostly, and sometimes when we wanted to get a lot of bats at once in a special effort, we did it also in cave exits. So we mounted them with the tags. They were either put on them using cement glue that lasts for between 1 to 3 weeks maximum, and then it drops off on their fur. But for the adults that we're not expecting them to grow more, we harness them with collars. Those collars include a weak link, so they will fall at some point. And of course, we tested these collars to make sure they don't harm the animals in any way on several individuals in the zoo. What we got is tracking duration from 1 week to 5 months at 8 seconds frequency, which is a lot of data to deal with. And every time, so for every individual, when we held it in our hand, then we also could take other measurements such as age, sex, forearm length, weight, the usual stuff. And at the end, we also had to give him something to thank his effort. And this is a bit of natural mango juice, which they love, even though you probably think they must consider it as their last feast, but after that, they are released, so all is good. This is the Hula Valley, our study area, and we are looking now on an image of the touristic area, where a lot of bats love to forage. It is part of the Great Rift Valley that goes all the way down through East Africa. And as in East Africa, it is considered to be a very important biodiversity hotspot. It's very famous for birders, especially the ones that love to see migration and huge amounts of cranes during the winter. It's mainly rural. There's not so many settlements around, and they are scattered all over the place. And the vast majority of the area is agricultural, different fields and orchards. And it's full, thanks to the orchards, but also thanks to people that are planting all kinds of trees everywhere, with a lot of fruits for the bat, including seasonal, but mostly exotic fruits that are not native. And we estimate the population to be around 2,500 individuals, give or take, and to be relatively stable. So that's true throughout the year. Okay, what do we see if we are looking on the movement tracks of individuals? If you're interested, I can later show you and zoom in and zoom out exactly how we identify different things on the map, because this is the grid that we are looking through, and through a lot of observation on the movement tracks of individuals who formulate a lot of our questions. So if you're interested at the end, I can show you how that program works. But in here, there's a few things that are already evident just by looking at these 5 individuals foraging for one night. And that is that, first of all, at least some individuals fly in very straight and targeted flights. Some of these flights between trees or between a cave to a tree are up to 20 kilometers long. And sometimes even more. It just goes beyond the range, so we can't really tell, but probably more than that. They keep coming back to the same trees night after night, up to 2 months. And an interesting fact is that they forage together, as you saw, but they, as far as we know and could detect, including in other labs, they don't fly together, not in pairs, not in groups, and they do that alone. And what I want to kind of emphasize at this point, and it's very clear from this picture, I think, is that not all bats do the same thing. And of course, there are individualistic movement patterns that repeat themselves. And we are very interested in to understand what basically drives these differences that are so evident. So the first question that we ask is what explains differences in individual movement, and our prime suspect is age, because age is related to experience, and you expect more experienced individuals to be more targeted, more efficient, and to fly in a very more efficient manner. So what do juveniles in the first year need to learn? So first of all, they need to know where to find food, right? And the second thing that they need to know is when is the best time to visit the food. So let's say that they can learn where the food is relatively quickly, but in terms of when, it's a bit more complex. So for example, this is the distribution of two of the main species, the predominant species in summer, the mulberry, and the predominant species in the winter. And you can see that if a bat has never experienced winter before, as juveniles do, then it will have to know how to switch between, for example, mulberries and China berries and anything in between. And that will, you would expect to influence its movement pattern to be more explorative and less targeted. So to test these predictions, so we tracked 84 bats, that was before the study that I talked to you about before, 65 adults and 21 less than 6 months juveniles, and we tracked them for more than 3 weeks. We took the raw data. I'm not going to go much into detail about that, but there's a lot of background work about cleaning and filtering and segmenting the data. At the end, we got a set of movement parameters that we thought were interesting to compare. One of them, there's an example here, is straightness. So you would take the distance between, in this example, the distance that there is, this shortest distance, sorry, between a cave and a tree in red, and the actual distance that the bat did on the way there in white, on the way back in blue. And you divide the shortest distance to the actual distance, you get a number between 0 and 1. And then you can say how straight was the line, meaning how efficient was the track, how targeted. As for the results, you don't have to try and understand all the numbers that are happening in this table. I'll tell you briefly. The answer to do juveniles move differently than adults is basically not really. And we were very surprised by this because, again, we would expect the juveniles to be more explorative, at least, than adults. And so what can explain this lack of differences? So first of all, pups probably learn very fast and efficiently to navigate. So they know very fast where to find relevant trees for now. Up to 90 nights since the first time they started foraging, they have very good knowledge of all the locations of the different trees. This is also recently published. But still, throughout the year, they would have to encounter novelty at some point. So it doesn't explain the whole thing. And you would probably also think that they probably can learn from conspecifics, but as I told you before, we don't see them ever flying together. So maybe they're observing other individuals, but if they don't follow them, how do they know where the trees are and how to fly there efficiently? So these are just still open questions. Going back to what explains individual differences, because age does not. So sex and reproductive stage do not explain it just the same as age doesn't. And now we're trying to look on whether individual personality, unrelated to the other factors, could explain individualistic movement. I'm not going to go into it in this talk. And looking on group level effects, asking basically, do different colonies move differently? So this is the main topic here, and you can see two caves in yellow and in kind of blue, I guess this is, 3 kilometers apart. And these are different individuals flying off this cave. And at the beginning, we wanted to ask whether these movement patterns are different, but then we realized something else, that maybe the movement patterns are different or not. I guess it's a bit subjective, but it's pretty obvious that these two colonies don't use the same space at all, even though they're very close. The yellow ones go up the blue ones go down, and they barely overlap. And so we changed the research question to something a bit different. Is spatial partitioning, this division of space, consistent first? And can we explain how it came to be? So we dived into the literature and of course found that we're not the first to ever ask this. So the seminal paper by Wakefield et al. Was done on northern gannets around the British Isles. And basically showing that colonies that share the same landscape segregate in space, as you can see here from the tracks. And there are, of course, advantages to spatial partitioning. It minimizes competition. It also sustains inter-colony information flow that is confined to a smaller area and within a smaller group of animals. This is simply because, it's easier to share information with a smaller group than a huge one. And so the easy case is where colonies are located far enough to passively create minimal resource overlap, as you can see here. But there are many examples of neighboring colonies that are located very close to each other that segregate too. And that aspect is more interesting, I think. One of the gaps that we found in the literature that almost all of the examples of this phenomena of spatial partitioning are very much focused on marine birds, which is not a problem with birds. We love birds, but most of these species breed colonially only part of their life. So it's not going to be the same thing for an individual that is a central place forager throughout its life as many bats are. There are few examples on other species and none to the best of our knowledge. I'm happy to hear if there's something new that I don't know of on bats. Okay, but maybe more interesting than the fact that we are selecting a different species is, can we explain the mechanism that stands behind this spatial partitioning? So the first candidate is a null model, basically saying that colonies by chance can segregate. We don't know really what means a neighboring colony. I mean, maybe the picture that we saw before the colonies are actually located far enough from each other, and distance constraints by their own could create partitioning. So that would be the null model. Then we wanted to add something more interesting. The most hypothesized mechanism that people argue throughout the literature is that competition, of course. When these individuals meet, they compete, and then they have to segregate. That makes sense. But there are two open questions. First is really, is it true? I mean, do these individuals even compete? So the general theory says that the colony grows. As it grows, competition within the colony increases, and then they spread to farther distances, and then they meet the neighboring colony's area. Competition increases again, pushes them back until the two neighboring colonies are actually separated on the line of equidistance between them. But nobody has really proven that when these colonies meet, density-dependent competition increases. Another way to look at competition is kind of like ants, where they maybe even recognize opposite colony members and fight against or compete against them directly. And there are some examples, at least in mammals, at least in mongoose, but it's less likely to be the case for bats. A third mechanism is memory, meaning in our case, a tendency to go back to the same locations again and again. And that by its own has been proven, at least theoretically, to separate between individual home ranges and also between groups. I think it's fursuers in this example. And the last one is conformity, and here I'll have to kind of explain what I mean by that. It's a form of information sharing, meaning that individuals share information within the colony, which makes them use the same subset of resources, which will potentially create segregation by its own, just because everybody forages on where he knows to forage, which is not the same places as the other colony, right? And of course, those things can act together. So memory and conformity could interact and create segregation by its own. It becomes very clear that in order to test this mechanism, you would need high resolution tracking data. Just to set an example, if you want to test whether memory influences the behavior or not, you'll have to understand what is the tendency of individuals to come back to the same locations. But if you have low resolution data, then sometimes you even miss visits to a tree, so you wouldn't know how frequently it visits it. And this is what we saw if we summarize all the tracks, at least for 2 main seasons. This would be winter in the left and summer on the right. And you can see that those 2 colonies separate in space, as we said before, and we calculated the overlap between them to be less than 10%. And you can also see on the pie charts on the left that, so not only do they separate in space, their tendency is to keep, so they are very faithful to their colony. So they keep coming back night after night to the same colony. You can see here for Gershon, the bigger colony, and 73% of the nights they came back to Gershon, and only 10% they moved from Gershon to Zema, so very rare occasions. Okay, a little bit about the methods. I will try not to be too technical here, so I ask for forgiveness in advance. So we calculated 2 indices of overlap. One is of trees, we call it resource overlap. So on average, 5.4% of trees were shared between the colonies, average across months, because months say different resource availability. And about 10% on average of areas, meaning if you take all the individuals visiting everywhere and you make a home range out of it, and you ask how much it overlaps, it's about 10%. So in order to compare it to competitive models, we ran a few simulations. First of all, we let a simulated bat on the computer, of course, go and visit all the available trees the bats actually had during this time. And we drew movement parameters from the real movement track. So for example, what is the distance that you're going to travel now? Let's sample randomly from the distribution of distances that actually exist in the data and give it that distance, and let's see which tree it finds, something along those lines. But then to answer more mechanistic models, more interesting models, we added to this null model different variables. For example, memory, which we'll explain a little bit soon, conformity and competition. After we ran each model, we ran it for 100 times, and we calculated those 2 indices, the resource and area overlap, and compared what we got from each model to what we have from the observed to see whether it reaches the same thing. This is basically it. Let's just give, and just a small note here. We wanted to do all of the things that I just explained, but we also wanted to know what will happen if I artificially take some of those mechanisms, for example, memory, and increase it gradually. Can I get to a value in which by its own spatial partitioning is achieved or not, which will of course mean that it exceeds this particular species behavior to maybe other species that don't have the same memory, for example. You'll see the results, and I hope all of this will be clear. So just as an example, we don't have to go into all the simulations, but essentially if a bat leaves a cave and it's not its first night, and we are trying to apply a memory model, then we draw a memory coefficient. So for example, for July in here, you can see that most bats, on average, 95% of the nights they visited trees they already visited in previous night. This is what the memory coefficient basically says here. And the theoretical coefficients are artificial, right? So I put 0.2, 0.4, etc. Based on what I chose to do. And if it was a memory step, then you have to go and select a tree you already visited, etc., etc. Okay, let's, even if none of it resonates with you because it's a bit complicated and I had to go through it many times before, then I'm pretty sure that the results would speak on themselves. You just have to believe me that the simulations themselves actually tested the different mechanisms that I mentioned. Okay, so let's see what happens. So if we look on resource or tree overlap, so this is the observed value, right, around 5%. And here on the x-axis, you're going to have the different models and to see whether we arrive to the same tree overlap or not. Okay, so let's start with the null model. In the null model, only distance or chance, we have 50% overlap, not even close to the observed. Then competition it's not very different from the actual null model. And if we add conformity and memory, it's very clear that memory, none of them really reaches the observed resource overlap, but memory is the closest by far. And also the interactions don't change much. So it seems like memory, at least in the species in this system, is really the main driver for partitioning of trees. of resources. We looked on the same thing with area, not trees, but home ranges of each colony. And we got very similar results, only this time we did reach the observed patterns. And you can see that conformity here with the tiny overlap is actually, this is actually statistically significant. Sorry, I forgot to write it on the graph. But also conformity on its own can reach the observed area overlap. Okay, so what all of this really means. Basically, we saw that resource partitioning is not a byproduct of distance from the roost because the null model was very far off from the observed. We also saw that competition is not a great contributor to partitioning, although it is theorized to be the main mechanism. And we also didn't find any evidence for competition when we actually looked on the data. So this is not a great way or the best way to look on whether competition exists or not, but trying to use our data and kind of get a glimpse into whether it exists or not. And we tested, for example, the number of bats on a tree and whether it changes the probability of a bat visiting it. Thinking that if there are too many bats, about 5, for example, the probability of it visiting a tree will be maybe lower if there is competition. And we didn't find it. I'm going to skip this part. And we also saw that memory is high in these bats. You can see the distribution of memory coefficients for both caves. And that could potentially explain why memory was such a strong driver of partitioning in the system. Lastly, conformity was also important, less than memory, and it could act on top of memory to enforce partitioning. This is at least what we think. Okay, now I'm trying to play a little bit with the actual coefficients, so of the different variables, to see what will happen if I artificially select a memory coefficient. And so in here you can see the different constant coefficients, and again the weighted tree overlap as we saw before. And you can see that in high values, memory alone again can reach the spatial partitioning that we observe. And that is, if you remember the part here in July, that is relatively similar to what we have actually from our observed data. Yeah, and the same is true for conformity. So conformity in resource and trees overlap didn't come as close as memory to the spatial partitioning. So we could have deemed it as unimportant. But actually, if we artificially increase the level of information transfer or conformity in our case, we can see that at high values, it too can reach a spatial partitioning. So imagine, for example, a species that does not rely heavily on memory like my bats do, but relies more heavily on information transfer, for example, but in the same magnitude, then only sharing information within the colony, so it seems, can also create the same partitioning. And it remains to be tested what type of information transfer, if any, helps to create partitioning. And the main candidates, which I'll explain more detail in a bit, are local enhancement and information center. And also it's interesting to see that we didn't find any additive effect really. I mean, in the interaction of memory and conformity, it's not a result that I'm showing here, but we didn't get different results, meaning each of these mechanisms probably works in isolation and not on top of each other, which I have to admit is a challenge to explain. But if you're interested, we have some interesting ideas in the paper, and you're welcome to go and check out. To conclude this research, so we saw an example of partitioning in wild animals that are not marine birds, so 1 point for bats. More importantly, personal and social information drive resource and spatial partitioning, which is one of the main ways to understand one of the fundamental processes enabling intercolony coexistence. In contrast, we didn't find any support for the existence of competition, despite the fact that it's so widely argued. And we argue again, by this point, I guess it's not a surprise to you, that high tracking data is really irreplaceable to answer some questions. And this one, I think, is another example of that. The next steps would be to see whether the role of information transfer and memory to group level processes as a whole and partitioning included into it. Maybe we'll open this Pandora box of looking on animal cultures, because now we know that the two colonies are moving to separate areas and don't overlap. So maybe they have other things that they don't share between colonies, and maybe they know some things that the other colonies don't, etc. And maybe it's even transferred into between generations. But this is something that we're still very far from even trying to understand. And as I mentioned briefly, we're looking on personalities within colonies. So imagine there's like a evolutionary stable strategy in which some personalities, the distribution of personalities is the same in different groups. So you can't have too many aggressive individuals. You have to have, I don't know, a few number of aggressive individuals to sustain a viable group, for example. So that's another thing that we are looking at. But in here, I will focus on the time that I have left on our attempts to understand how information is transferred within the species. Okay, so let's first ask the main question. Obviously, these bats really like to be together, as many other bats, I assume. So what is the advantages of congregating in such big numbers, either in roosts or trees? Of course, it decreases predation and parasite load. It's useful for thermoregulation, even though this species does not hibernate, but still in cold winters, they really like to be together. Actually, also in very hot summers. So maybe that doesn't explain it very well. And of course, it has social and breeding benefits to be together and exposed to each other all the time. But in the context of this work, we're interested in we're interested in sharing information. So of course, living together and sharing information kind of saves the energy that you would need to go and explore resources alone. It reduces uncertainty about finding food, because if you didn't find anything, maybe your friend did, or your colony member. And it improves in general the breadth of information that you have about your home range and the quality of information, because you're not sampling it alone. Other individuals that have the same motivations as you sampled it too. But of course, it also increases competition and social dependency, and there are a plethora of other disadvantages to live in this way. We were interested in checking out basically two main mechanisms. One of them is mechanisms of information transfer. One of them is local enhancement. So basically what you see here is a naive bat in blue, leaves a cave roost and tries to see where to forage, where there's one profitable patch with a lot of bats and others with less bats or none. So in local enhancement, the bat goes and explores, then it maybe arrives to a detection range where he can hear, or in this species see, a lot of individuals on the tree, go down there and eat. You know, you have to consider that this kind of movement will demand some level of exploration, but once you found a good tree, you just keep coming back to it. And so the exploration is very specific. The other maybe more interesting mechanism is information center that we know that exists in maybe at least colonial breeding birds. So a knowledgeable individual here in red comes back with a new fruit on its mouth to the cave. Then in the cave, it's not clear, but maybe the bats smell it. Maybe they can even socially communicate to deliver some type of message. This is also an option. And then the naive bat learns that there is something new, and then it will probably have to follow it back to see where this new food is. But as I mentioned before, we didn't see any evidence for following in the species, so we were really interested to know whether they can still do it without following each other. Just a brief note, the information itself does not have to be very individual necessarily. It could be that it just came back with a lot of smell of a new fig, for example. Everybody smelled it, the information is public, and then everybody knows, or knows who to follow later. I will just skip here a little bit, basically saying that we know that resource predictability really shapes information use, and this is a very cool paper by Egerberg et al. That shows this on 5 species of bats. So basically on the right, Myotis vivesi, you have the fish-eating bat, you have it feeding on ephemeral resources, right? Fish are not predictable in space. And on the other side, you have a species that feeds on beetles, and it's considered to be more predicted because these beetles are probably found in more or less the same location, at least compared to fish. And you can see that the individual on the right, the one who uses unpredictable food resources, is much more explorative. And maybe more interesting to our issue is that it also is more prone to use social information. So in here, what you see is that the fish-eating bat is listening to calls from conspecifics, approaches them if they have searching calls, and even more so if they emit feeding buzzes, while the other myotis does not at all, is not at all influenced by what he hears at all. Why is it meaningful in our species? So, of course, because they eat fruits, you would expect that the food will be very predictable and there will be no need for information transfer. But I'll try to argue in the two next slides that it's a bit different than that. So first of all, so this is one of the typical experiments that we did actually to ask a whole different question about navigation. So we translocated individuals from the south area and released them in the northern area of the home range. And as you can see, they move very straight, very fast, either to the tree they visited the last night or to the cave. So that kind of tells us, it's a hint to the fact that bats know very well where to find food from everywhere you put them. And this is a tiny example to show the straightness value that I showed you before. Also in those translocation studies, it's very high, more than 0.9, and even higher, a little bit higher during routine flights. But if you want to hear more about navigation and why do we think, no, almost, that bats have a cognitive map and can navigate very well, you have to stay tuned and to hope that the video is going to come to give a talk soon, and then you can hear all about that. We also know that even though they know where to find food, it's very difficult to know when to visit the fruits. We talked a little bit about this before, but the best example of it, and this is the focus of this study, is the Ficus sycamorus, which has about 18 cycles of fruits a year, so it's very, very unpredicted. Due to time, I'm going to skip a little bit the introduction, if I may, and just jump right into the results, okay? I don't think it will be a problem. Okay, so basically what we tried to do is we ran field experiments to check for information center hypothesis, once by spreading fruits in the cave when the bats were not there and seeing whether they smell it and go to this Ficus sycamorus tree. We have only 2 of them in the field area, so it's very easy to see when the bats fly there and not to any other tree. And we also wanted to see whether the information is maybe transferred in an intentional way by smearing bats with the same smell of the same food that we had in the freezer, releasing them at the end of the night as though they came back from foraging night, and seeing whether naive bats go to visit that tree or not. And we also looked at local enhancement by emitting sounds that we recorded in a cave on trees without fruits, and seeing whether bats that fly nearby descend and go and visit that tree. I'm already going to tell you that the smell in the cave without smearing it on bats did not work. Maybe for technical reasons, maybe for real reasons. And this is the result of the information transfer experiment. So in here on the left, you can see one night before the manipulation, you can see the Ficus sycamorus here in the north. This is me driving in gray with a smeared bat and releasing him in the small cave over here. And this is the movement that they did during this night before they met this smeared individual. And then this is one night after the manipulation. You can see that the orange naive bat visits this Ficus sycamorus, so apparently learned something new. And also, surprisingly, the smeared bat itself went to visit the same tree again, which he should know where this information is coming from, but I guess he was curious. And interestingly, 2 nights after, also the blue one visited the tree. Why I'm saying it's interesting? Because they didn't roost in the same cave, as you can see here from this picture. So what happened here, we think, here marked in red, that these 2 individuals, the gray and the blue one, they met on a tree. So maybe information center is not necessarily in a cave roost, but it can also happen in a cave. Okay, I'm going to skip the local enhancement experiments, if I may, and just jump right into the discussion. So we find that high throughput tracking, as in the Atlas system, together with field manipulations, which is a great privilege that we have to use, can shed light on long theorized mechanisms such as information center theory in wild animals. It gives evidence for both the information center, and you didn't see it, but trust me, on local enhancement as well. And we have, of course, many different questions that we want to ask after this. And I think just because of time, I will conclude here. And I will just say thanks for your attention. And also, of course, for all the lab members and helpers that I had along the way, and for the funders of these projects. Thank you very much.
Thank you, Emmanuel. That was amazing. It's just brilliant to see the amount of work that you've done and the such detailed information that you can get with the tracking devices that you're using and what we can learn about the bats based on that. So thanks very much. That was really, really interesting.
Thank you.
Let me look. I think we have some questions or comments in the chat. Moira says, the ways and means of studying are so complex. This is fascinating. Looking at your hypotheses and the way you formulate them and the conclusions you come to. So yeah, I guess we all agree.
Thank you very much.
With that one. Isabella says, thank you, Emmanuel. Great talk. Question. You know the bats really well. What result were you surprised by the most so far?
I think there's a bit, thank you, Bella. Good to see you. I think the most surprising thing, and this still remains an enigma, it's a double enigma, I think, that kind of comes together. First of all, everybody says this species is very social. And we know that it has a stable producer-scourger relationship in which they even feed from each other's mouth, etc. But they don't fly together, almost ever. And you would think, okay, so what is the point of technically being only social during in the roost, but then going to other trees and not flying together at all? And I'm very surprised to know that they can share information and they communicate a lot, but don't keep the same social contacts probably, and don't use it to actually see each other and follow each other in the field. So that's something that I would very much like to know the answer to.
Yeah, that's great. I was also really intrigued by the fact, well, not intrigued, I guess just amazed by the fact that there's so many different ways and reasons why the bats transmit information to each other.
Yeah.
And it's,
imagine how it is on in the saurian species that have much less predictable food resources, that they really have to share information all the time in order to survive.
So that's one question I had actually. How unique do you think this species is, or how kind of wide they're representative of processes that might be going on with other species?
That's a super good question. So it is unique also because of the environment, you know, it's not in the tropics, but I would argue that a lot of food bats basically have to deal with the same challenges, right? Even if they roost on trees and in smaller groups, they will still have predictable and unpredictable foods to eat from. They would still have to kind of balance between memory and social information, maybe to different extents. I think that at least the methods apply. I think if you could track other species in the same way, I guess you could answer similar questions. Whether you see the same behaviors or not, without doing a proper research, I don't know how to answer that question.
Lots more work to be done, I guess.
Yeah, there are new systems and tracking of the same reverse GPS system. At least I know of one in Mauritius on another food bat. So let's stay tuned and see what happens there.
There's a question from Eleanor. She asked, on a practical level, I assume you glued the tracker between the shoulders. Did it not affect their flight at all?
No, so that we've seen so many times. We tried different methodologies at the beginning and different ones later. We checked it also on zoo-kept bats to see whether they can move normally. And we also looked at many movement behavior afterwards, and they all look similar, no matter if we put it a little bit up or a little bit downwards. Of course, you won't put it on its head. But other than that, they're very easy to work with animals, and they're very flexible and adaptable. Whether it troubles them or not, I honestly can't say to what extent, but I know they behave naturally. And this is our index of knowing that everything is fine with how we put those tags. But it's an important question. Thank you.
I also wanted to ask you about, I found one of the findings that struck me the most was the fact that you didn't find any evidence for competition driving resource partitioning, which is kind of a big assumption in ecology in general, not just for bats, but for many species. So yeah, I just wonder if you could comment on that a bit more, on the fact that didn't really, your results didn't actually find any evidence for that.
Okay, so there's like an interesting and technical answer to this. I think the technical answer, which is less exciting, is that we didn't observe competition. It happens in very brief occasions. We didn't measure it correctly. It actually happens all the time and we can't see it. For example, because we're using a subset of individuals, only the tagged ones, and we don't know what happens with all of the other bats. So maybe there's a lot of agonistic interactions where they meet and we miss it. I don't think so, because we have really a lot of individuals by now that we tracked, and we didn't see evidence for it in other systems. But that's like the technical answer. The more interesting thing, I think, is kind of thinking about the evolution of how these colonies came to be and to share the same space. And I think probably competition was there only in the initial phases. So when they have to form, right? So everybody's exploring the new territories. One colony grows until it's too big and it goes to another neighboring cave, etc. And then they figure out who goes where, kind of, with competition, with social information, much more abundant and strong. But then after everybody knows its own range, and it can, as you saw, they really rely on memory to, and also social information, then, there's no need for conflict anymore. And they can just basically, like, we draw borders between countries, not that it helps us, but, that's the main idea.
That's great, really interesting. There's another question. Does climate warming changes affect your bats and the food availability? And I guess following on from that, would that affect resource partitioning and the mechanism?
Interesting. Well, I guess a catastrophe will change everything, I suppose. I mean, if one colony is really deprived of all its resources, then a new balance will have to come about. And I'm thinking about fires, or in our case, it used to be a swamp. So a huge, flooding of everything that will sink all the trees in some areas and others. Maybe something like that will, of course, affect everything. But what we are also seeing from the movement, that they are very unresponsive to changes, at least in weather. It doesn't mean whether things will be very extreme, but in Israel it gets super hot, like 40 degrees Celsius outside. In the cave it's not much more pleasant. It's even 100% humidity. Some winters are very cold, and in here, because it's a valley, winds are so strong, and you just see them fly regularly as though nothing happened. So I think they are, I don't know if it's okay to say it here, but badasses, and they can really deal with different conditions. Whether the situation will be much more extreme, it will be difficult for all the species and them as well. And I'm sure it will affect spatial partitioning, just because it will affect the abundance of resources. Now everybody has enough resources to forage on. If that changes, then the rules of the game change as well.
That's great answer. Thank you very much for that. I think we've come to the end of our 1 hour session, and I can't see any more questions in the chat. There's lots of comments all of them very positive, saying fascinating work. Thank you very much. All very exciting, lots to think about. So yeah, I think everyone has enjoyed the talk as much as I have, and I've certainly learned a lot. And I'm really looking forward to hearing more about your work, looking at some of your papers to kind of dive into the results in a bit more detail, because it's super exciting, and also the technologies that you're using, which, as you say, can be applied to other species to learn much more about them. So thank you very much.
Thank you very much. Yeah. Thank you, and you're all invited if you ever want to come. It's a beautiful area in Israel, and you can see them in your own eyes. So you're very, very welcome.
Thank you. That was brilliant. Thank you, Emmanuel. Thanks everyone for joining. And yeah, please keep coming back to our webinars in the future. Thanks very much again.
Thank you very much.
Have a lovely afternoon, everyone.